पूछ रहे थे। Good morning, all the participants. We have with us our speaker for today's session, Dr. Vijay Singh, sir. Sir, I can see you. Your video is available. Sir, your voice is not audible. Sir, your voice is not audible. Please check your microphone. It's on mute mode. The bar in between.
Yes, can you see now?
Yes, sir.
And about PPT, can you see the PPT?
Not now. Sir, you have to present, you have to share your PPT first.
Yeah, so I'm going to
present now.
Present your entire screen.
Yes, sir.
And share. Somehow it is not clicking. Wants to share the content of the screen will be visible.
Sir, you have to click on the screen, the pop-up window. Just click on the screen, and then the share button will be highlighted.
Can you see now?
Yes, it's now opening. Yes, sir.
Okay.
Sir, just keep it on full screen mode.
Yes.
And sir, please hide that bar which is coming, asking to share.
Done.
Yeah, perfect, sir.
Mm-hmm.
Now, may I ask all the participants to please put their mic and camera on off mode, mute your mic, put off your camera. We have with us Dr. Vijay Singh. He's Joint Director at Food Safety and Standard Authority of India, Government of India. Dr. Singh did his PhD from University of Pune in metabolomic syndrome and asthma. He was serving as veterinarian surgeon at Sanjay Gandhi Animal Care Center. He was veterinary officer at Government of Rajasthan and then at IGIB CSIR New Delhi. He served as assistant professor in Academy of Scientific and Innovative Research at IGIB New Delhi. His expertise is in social and behavior change, animal ethics, animal welfare, dairy husbandry, and milk processing. Currently, he is working on food safety, antimicrobial resistance, veterinary drug residue, quality assurance, and good lab practices. Dr. Vijay Singh sir will be delivering his lecture on design of animal experiment, welfare, and necessities. I welcome you, sir, again. Over to you, sir. Sir, you can start your presentation now.
Okay, good morning, everyone. And first of all, I would like to thank Professor Anjali Avasti and Dr. Kumud for giving me the opportunity to interact with you all people from the Rajasthan University and from other university. And today my talk is about design of animal experiment, welfare and necessity. As the series of this webinar is on basically the molecular techniques, and you might have studied about the proteomics and all. So I want to focus and tell you that we are doing most of our research perfectly, very good. But when it comes to translation to the human, you have to do animal experiment. And while doing animal experiment, we sometimes become casual and we don't do the way it is to be done. So I will today focus on experimental design and how your experimental design can affect your molecular thing, which will ultimately lead to the non-translatable or non-reproducible data. So let's start with first. So basically, I will start with the first. This the brain is a wonderful organ created by God, and it starts working as soon as you wake up. But we don't have a classroom. It stops as soon as you enter the lecture hall. So those of you, I may not see you. I cannot see you all because I have full screen with the PPT. So please don't go away because there will be exam after, and some few questions will be there which will be very tricky after this presentation. And you can very well ask me the question after this presentation and anything that you would like to suggest. So be with me mentally. First, what is an animal experiment? Dr. Anjali, just want to confirm. Can you hear me clearly? Is it
Yes,
okay?
sir. Yes, sir. We can hear
Okay.
you. Perfect.
Okay. If anything goes wrong, just tell me because
Sure.
I cannot see anyone.
Sure, sure, sir.
What is an animal experiment? Basically, animal experiment. What do you qualify to call animal experiment? Is that one which is performed on living vertebrate. Living is important here. If you are doing something on tissue which is brought from the slaughterhouse, it is not living. If you are doing on non-vertebrate, like if you are doing experiment on Drosophila, that it is not an animal experiment because Drosophila is not a vertebrate. It does not require any clearance from any ethics committee. Second thing, it's caused some distress or suffering to the animal. Even suppose if you are just taking a little bit of blood from the animal for their health monitoring, not for some of your experiment, it is not causing much suffering. Hence, in such cases, this experiment will not be called as animal experiment. It's done for certain purpose. Very, very important. Why it is important? That you have to justify when you are doing an experiment what is the purpose. And purpose cannot be your master degree, purpose cannot be your PhD degree, purpose cannot be that you want to gain a skill. Purpose is that you are going to solve a societal problem. And the harm that is caused to the animal because of experiment is much less than the benefit which you will get. So you have to first find out the harm and benefit analysis out of it. So three components performed on living vertebrate, cause some distress or suffering to animal, and is done for certain purpose. Now coming to the welfare, you might be hearing this word welfare a lot. And welfare is most of the time is being considered in our country as a wrong connotation that you are going to get something in free, like for Ministry of Farmer Welfare, means they will get something free, some urea, some kind of a subsidy or whatever. But welfare is a scientific term. Okay? And what is welfare? Welfare of an individual is its state as regard its attempt to cope with an environment. Means if you are put up in some environment, whether you can cope, whether you can survive or you don't survive, like we are all house, we are blocked down into our house. So we are living in a condition. We sometimes can cope or we don't cope, then we will have a problem. We will have the mental issue. We will have depression. If we can cope, we can survive and we can get weight. We can live happily. So you put animal in certain condition, whether animal can cope, can survive, or don't cope. Don't cope means don't survive, and they feel bad about it. Welfare, another issue that you sometimes say the universities set up or sometimes in pharmacy college that we are providing air condition, we are providing feed and water. What else you want for animal? So the welfare is having on COP is having control both mentally and bodily. I want to quote you example that if suppose I am being put in a jail, I will get everything, but what I will not get is the mental freedom. So this component of animal to get mental freedom is always missing most of the time. Sorry. And how can you provide that? Mental freeness and mental control. Suppose you have to enter to the mind of the animal, what animal really want. Like if you have a mouse, what mouse want? They want to dig, they always want to chew, they always want to clean themselves. So for that you have to provide the tissue paper. in the gaze of the animal so that they can make something, they have something to do, and they can say have a mental control. So bodily control is something like they can eat, they can do this and that, but mental control is like they can also do something out. Welfare always refer to the live animal and including humans, and it is the characteristic of an individual animal. If animal is dead, then there is no welfare. Welfare, sorry, welfare including suffering are a part of it. When we call welfare, it is not a charity. Welfare means even if you are a non-vegetarian, you want to eat chicken. Welfare means how the chicken is grown, how the chicken is being transported, how the chicken is being slaughtered, and like that. If you are doing experiment, welfare means how the animal are kept, how the animal are treated, how the animal are being relieved from the pain. That is the welfare. And welfare is extremely important for the animal experiment, and that I'm going to do. Animal protection and animal welfare is different. Animal welfare is the characteristic of the individual, whereas the protection is something human do to them. I give something to humans, sorry, so to the animal, and I protect animal. That is not welfare. Welfare is something what we give, what they need, and every animal need is different. How it is different? Suppose if you take a pig, what they want, they always want to go to the mud and they want to root. If you take the chicken, chicken want to roost and they want to fly. If you go to the mouse, mouse want to dig the hole. So every animal need is different. And what we always feel for humans, human always feel whatever we are providing, whatever is good for human is always good for animal. This is most useless idea. No, whatever is good for us is not good for animal. Animal have their own need. Animals have their own requirement, and we cannot say what is good for us is good for them. Coming to the value of animal, extrinsic and intrinsic. When you call the extrinsic value is what are we going to get from animal? Like from the buffalo, we get milk. From the animal experiments, the animal which are in your lab, what we will get? We will get data, we will get blood, we will get tissue. Out of that, what we will collect? We will collect the data. So that is the in extrinsic value. Extrinsic value is our PhD data we will get from that or our master data. And that extrinsic value will only be good when the intrinsic value you will provide. And what is the intrinsic value? Intrinsic value is the value of animal. Intrinsic value of Vijay Singh is intrinsic value of my value. And nobody can reduce and nobody can increase my value. Intrinsic value is always fixed. Intrinsic value is my inherent worth. And it excludes utility. Utility means something which I give out or some animal something gives out like food, like data, your milk, your leather, your meat and all this one, that is extrinsic. Intrinsic is what is their inherent worth. If you cannot give the intrinsic value good to the animal, extrinsic value can never be good. So, if you will not provide me the good housing, good conditions, good food, very good means outside conditions, I will not perform well. If you want your employee, if you want your student to perform well, you have to always provide intrinsic value good. Then only they will provide their best. Hence, if you house an animal in your case, remember to provide what they want so that they will provide you the data which is reproducible, usable, and also which is translatable to the humans. Now coming to the validity of experiment. Experimental validity is something like the validity of your driving license. If your license is not valid, policeman will catch you and put you fine. Same way, there is a way to check the validity of experiment. There are two types of validity: internal validity, external validity. Internal validity is something where you can establish cause and effect. What is that cause and effect in simple terms? You give paracetamol, temperature will go down. And you make sure the temperature which is gone down. If I have a fever, okay, 102, I will take paracetamol, temperature will go down. I have to make sure that my temperature have gone down because of paracetamol, not because of the mother, this dalia, or I have put something cold water on my head or anything else, on any food or any luxury or by air conditioned room. My temperature have gone down because of paracetamol. If you can establish the dependent and independent cause and effect way, your experiment is internally valid. Now, external valid. When you have done experiment on mouse and you have an interesting finding, and you say, I have discovered drug for the diabetes, I have discovered drug for the cancer, I have discovered drug for hypertension. External validity is that you have done X drug discovery in X species and strain of the animal. There are more than 2,000 strain of mouse. But what is the potential of this translation of this research which you have done mouse in human? External validity is if you have done in mouse, what are the chances it can be replicated into dogs? What are the chances it can be done into humans? What are the chances it can be done in other species? So internal validity cause and effect. External validity when you can translate the same result in another species. So it is very, very important. How can you make sure internal external validity? You can make sure internal external validity when you select the real animal model. And real animal model does not mean the model which is available in your animal facility or any mouse or any rat. Internal validity, external validity means when you select a model whose physiology and pathology construct validity, whose changes are same as like which is same in humans. Like I tell you example, we are doing a lot of research on diabetes and in mouse, but the basic site of glucose clearance in mouse is liver, whereas the site of clearance of glucose into the human is skeletal muscle. There is a big difference. So if we know the difference, we can tell, oh, we have done it mouse, but we can. So internal validity is maybe high, but the external validity is low because the site of glucose clearance in two species is different. Now coming to the history of animal welfare. As you know, in our country, we treat animal as a god, goddess, and I have traveled all over the world, but the way we treat our animals mostly, I'm not saying everyone, but mostly it is very nice and we treat them well. But the first documented history of animal welfare is that when Bramble in 1965 published a report on five freedom. So people were using animals, they were feeding them, and then they were butchering them and eating them. Then some of people were slaughtering the animal, and by the time the animal reached slaughterhouse, the animal used to be in really bad condition, like cattle and buffalo, and not buffalo, but cattle. So they decided, okay, we should have a welfare for the animal also. And then they found out that it is not only I told you, the bodily control, mental control. It is not only the bodily feed. If you feed them, they are growing and they are breeding. Does not make sure that they are having full welfare. So he gave the concept of five freedom, the list of five freedom, that freedom from hunger, freedom from thirst, freedom from injury, freedom to express natural behavior. But these five concept is also becoming outdated, and these all now welfare is becoming an obligation in relation to causing harm or poor welfare in other animal, and the assertion of right and freedom cause problem. We sometimes say it is our right on animal, but we have also duty that in our constitution article 51 they say that we will treat animal with a compassion. So assertion of right is also sometimes cause problem because animal also feel pain. And the basic concept that animal feel pain, any procedure, remember, any procedure which cause pain in human also cause pain in animal unless it is proved otherwise. So, and another important thing is that sometimes they don't, if I have a pain now, I will start growling and making noise and howl and run and go to the doctor and hospital and get the injection. But not necessarily animal express pain in that way. Most of the time we say researchers, they say they want to collect the blood, they cut the tail and take some of the blood and say, oh, they are not making any noise and they are okay, they are feeding, they are taking food. Are they not feeling pain? They are feeling pain, but we don't understand their pain. So history of the welfare is that it started with Bramble, gave the five freedom, and now we have come to the find where we can measure welfare. Welfare is something not in the air. Welfare can be measured. How it can be measured? By physiological indicators of pleasure. Physiological indicators are like your heart rate, your respiration rate, and your skin temperature and all that one. Behavioral indicator of pleasure. Behavioral indicator like if you go to the temple and if you sometimes see the animals, elephant are doing head like this means stereotypic behavior, purposeless behavior, doing the same time without any purpose is a stereotypic behavior which is indicator of poor welfare. Extent to which strongly preferred behavior can be shown means how they can show their preferential behavior. Sometimes the animal are put in the cage, like chows are put in the chow crate, and chicken are put into the battery cage, A4 size cage paper, where they cannot express their natural behavior. So, variant of normal behavior shown or suppressed. Extent to which normal physiological process anatomical development are possible because of the housing condition. Extent of behavioral aversion shows means I told you the stereotypical behavior and biting of some of the bars which are there in their chow crate and all. Physiological attempt to cope or fail to cope. Sometimes they start biting their own tail and they bite their tail so badly that their tail is gone. Immunosuppression. They are so much depressed that their immunity is suppressed and they get the disease. Disease is more. Disease prevalence increase in your facility or in your herd. It means the welfare is poor. And also the behavioral pathology, brain changes, body damage prevalence. You see some of the scratch, some of these wounds outside the body, which is indicator of poor welfare and reduce ability to grow or breed and reduce the life expectancy. So, welfare can be measured physiologically, pathologically, behavioral wise, and also molecular. If you see the beta casein, I mean, some of the enzymes which are cortisol, if they're cortisol and in the human and corticosteroid into the animal, if they are increased, it is a clear cut sign of the welfare problem. If the pain receptor sorry, enzyme P, which is indicator for pain, increase, it's a clear-cut sign the animal is pain. So we can very well measure scientifically by different tool that animal is in pain and the welfare is poor. Here you can show the chows, which are throughout life, they will be in this chow crate and they are biting because they have nothing to do in their whole life. They will be put inseminated from the back by the artificial insemination and you can see the dog which is put into the cage which is showing you very least interest. In such case, we can say the welfare is poor. Why should we do welfare? For animal experiment, people say that we have so many humans and cows and dogs and on the road. Why should we care about the animal which are there in our case? Because first of all, you are doing research. You are publishing. And if you are publishing, you are making the basis for the research for other people. Other people make assumption on your data. So if your data is not correct, if your animal is not put in a good welfare, you are giving a wrong basis for other people. Another thing that if you are doing research not for the animal, sometimes for animal, but most of the time for whom? Humans. If welfare is poor, if the welfare is not taken care, if the animal model is not right, this is going to come to us. Like I give you an example, TG1419 exam recently, so it was gone through the mouse, through the monkey, and other animal, but still it had a very big problem into a France study where the study was from a sponsor was from Portugal that the humans were had a very serious issue. So it is because of this reason we are doing research for one, humans, and also animal. Also because we know, sometimes we don't know, but we know most of the time that animal have the same capability as like we. So I am going to prove this. So why do we welfare? Because we are doing for us. We are doing this data for whom? Human. Now, the concept of anthropocentric view that we are central, we are the best, and what is good for us is also good for animal is not true. Animal have their own world. And this is proven by the almost all variability reported in primate can also be found in fish. So fish is as intelligent as the primate. They are the fastest. Sometimes they can evaluate fast and also respond very fast, like a blowpie. Come and sit and immediately go back and fly again because they don't find they are very slow in responding. Humans are very slow learning. Number of error in 5-year-old child, cow, sheep, goat, and pig perform less well than the dog, cat, rat, horse, etc. So this is way that when you compare the 5-year-old child with other beings, it is not always that humans do the best. They can also discriminate and recognize. If you go to the sheep, I will give you the example in detail how they are judging us by ear position and also by the eye. So they can recognize and also they can discriminate. They are also very innovative. Like if you give a piece of bread to the crow, they will not eat. They will go and put into the seashore, attract the fish, and they will take the fish. So they are innovative. They have the idea. They also have the capability of, like, if you have a group of heifers, means young buffalo calf, and means which have the potential to go and eat directly. So there is a group of heifer we can go and eat, and there is a group of heifer which have to press the panel, and then they will have access. Generally, people will answer in my classroom when I give this talk that what is will be preferred by heifer. Everybody says that something which they have direct access, but no, heifer like when they do something and when they get the food thereafter. So they go jumping, their heart rate is so they are happy. So they want to do something before they want to have access to the food rather than having direct access to the food. So they have such capability, feeling and emotions. So in that case, when they have such emotions and capability and feeling, we should treat them well. Like pain, in the pain, dog vocalize, but sheep don't. If the animal is in pain, I'm in pain, dog is in pain, they vocalize, they cry. Sheep, sometimes even if you cut the part of a sheep, that some surgery is being done, like in the Australia, to cut out the part so that they don't attract the fly, but they will never make noise. Though they will express in different way. They will express their gait will be different. Their cortisol level will be extremely high. Does not mean that a dog which vocalize feel pain, a sheep which don't vocalize don't feel pain. So feeling same and response may be different. Even if you put sometimes some people who are eating fish, like they think that fish is like another vegetable. But do fish feel pain? Yes, they feel pain. How do you prove they feel pain? If you put a acetic acid in the mouth of a trout is a part of sheep, kind of sheep, and they always just shown the sign of pain. Okay, you put acetic acid in their mouth, they will show the pain sign, and the sign of pain will be gone if you give morphine. Morphine is nothing but painkiller. So it means experiment have shown that they feel pain. They also feel guilt, anger, and rage, like dog. If you have a pet dog, you offer some food and you tell dog not to eat this food, and you go out of the room. And after coming back, if suppose your dog have taken that food because it's too tasty and you have gone out, when you will come back, sorry, when you will come back, you will see that dog is realizing that they have done wrong, and they will also feel guilt and anger, and their guilt and anger can be how they can be monitored by heart rate and some other parameter. So there is a way to monitor where guilt, anger, emotion, welfare, and everything. And welfare is not always same. Welfare is different. So I want to give example like pre-parturient, just before giving birth, shall prefer what most of the time straw. Straw means this raw part of our what we call in our Hindi term is bhusa or tudi. But if they give birth, what they like most? They like most food. Before the parturition, they were mostly busy with access to the straw because they need food. So these are the examples that welfare is not always same. And the welfare is gone poor because of our lot of genetic modified animals or genetic improvement is happening. Like earlier, you might have seen those who are eating chicken, that earlier broiler used to grow to 2 to 2.5 kg in 12 weeks. Now the same broiler is ready in 30 to 35 days. Okay, and this was just before 30 years. A cow which used to produce only 4,200 before in 1957 now is producing is 9,000 kg, and it is even more also now. So the genetic modification which is happening, fast growth which is happening, is not certainly good for animals. So welfare is poor in such kind of animal. So physiological measurement in relation to behavior, take account of behavior in interpreting heart rate. How do you measure welfare? So I told you, if you have a sheep in your this we call it this head, and you see how they react to the different stimuli. You can also read the paper. If you just put them separate, so there is no change in heart rate. If they are standing into a tractor outside, stationary trailer, no change. But if they cannot see visual isolation, they have a problem. So their heart rate increased to plus 20. If they introduce to new flock, they also have a problem, but problem is solved after 30 minutes. So the heart rate increase. If you introduce to the new flock, then there is a means 30 to 120 minutes. It increase to 30, then become 40. If you transport also, that is a big stress to animal, the heart rate is increased. If a sheep is being approached by a man, their heart rate increased by 50. But if a sheep is approached by a man with a dog, then sheep is really in trouble. Their heart rate increased by 84. So we can say by such physiological parameter that the heart rate is increased, so the welfare is poor and we should take care. By this graph, I want to say that even if we have to kill animal for our experiment, which I am not saying against, but kill in a way so that they don't, intensity is less and duration is short. So you have to adopt those parameters, those measures like giving injection of thiopentane sodium, which can quickly kill the animal, or by putting the animal into the chamber of carbon dioxide so that they don't feel pain, and the death is painless and also quick. So this is a way that we have to do experiment rather than prolonging the suffering as well as the duration of killing, which is not a good example. Do we all have the same idea of welfare? No. So we have done a study where we were discussing with the student, what do you call the welfare? So mostly the VATs and the non-VATs, this study was done. So when you consider VATs, VAT particularly consider though, welfare is means they are only when they are healthy. They don't consider it as mental, but mostly other way. When we ask the physical, whether welfare related to physical availability of feed, availability of cage, availability of water, then more or less everybody agreed, yes, these things should be available and it is a part of welfare. Welfare is also something naturalness, means you give the natural condition in that way. And many people also said. So there is a variation. Bats consider if animal is healthy, veterinary doctor, then their welfare is very, very good. Whereas other people think that yes, welfare is also related to also means their mental status. So this is do we all have the same idea? No, we don't have same idea. Why do we need welfare? We have already discussed. Better welfare equal to better science. Most research papers that simply say something along the lines of animal are housed in standard cage with softwood bleeders and this blah blah blah, with no explanation why animals were not provided with anything else. Animal studies most often not neglect the basic enrichment strategy, and hence experiment variability increase due to such negligence. So I give you example. If you house a dog for your toxicity study, some of you might have seen that dogs are this breed of dog which is beagle is being extensively used for. If you put that dog into a condition where they cannot move in the slings, and you take intermittently blood and you also feed the toxicant which is there, if you keep them immobilized into the toxicant in this slings, and another way you let them go outside, have a walk, take a walk, go inside, outside, their parameter is more reproducible. Another example that you have two sets of animal where on the right side where the animal were given means enrichment. So fibrillin 4 is a protein that is excreted by vascular smooth muscle and it is essential for maintaining arterial integrity. Homozygous sulfur knockout mice die before birth from the arterial hemorrhages, but heterozygous only appear outrally normal. So homozygous die, heterozygous survive. Although gaps are present between smooth muscle cell in the aorta and endothelium, also shows sign of damage. So the sign of damage, if you are providing with enrichment or without enrichment, is different. So in a controlled study, I was found that housing of heterozygous fibrillin 4 mice in large cages with a tunnel and wheel significantly reduce the gap and maintain the integrity of endothelium. So if you are providing them the tunnels, wheels, that is the enrichment. So you can see the picture on the right side, the gaps have reduced because the environment is enriched. If you don't provide that, the same condition is exaggerated. You can see on the right side, which is marked with the colorful arrows. So, as this webinar is about the molecular technique, so I am saying you are not providing something to the animal, it's reflected into your molecular aspect that can be visible through the molecular parameter, like I told you, the corticosteroid, and also visible through the histopathology, which I have shown in your arterial hemorrhages. And this is because environment factors such as housing condition also affect. So another example is that when you are, second example is when considering the effect of enrichment environment or level of hormone leptin on signaling protein known as eicosanoid and on the pro-inflammatory mediator COX-2. So these pro-inflammatory mediators, if you are not providing them, the enrichments are more and more, means pro-inflammatory environment is increased, whereas increased inflammatory state, if you give the standard condition means no enrichment and no taking care, but this tumor weight was also significantly reduced, pro-inflammatory goes down, COX-2 positive also goes down if you are providing them housing, enrichment and welfare. Now coming to the considering animal, what they want and what are we providing. Animal are nocturnal animal. Mouse is a nocturnal animal. Nocturnal animals means they are active in the night. But we are doing experiment most of the time when there is a daylight or there is a light in our animal facility. So it is like you ask me to get up at 2 o'clock in the midnight. Anjali professor asked me to give me the seminar at 2 o'clock and you all people come at 2 o'clock. How much will be the translation? Because that is not my active phase. I will come, I will be down, I will take coffee and tea, but I will not be so natural because that is not my active phase. Same is happening with the animals. They are normally nocturnal and we are doing experiments when there is not their active time. They are highly dependent on smell and scent. They like smell and scent. And if you go to the animal facility and you see, oh, it is smelly. No, it is not smelly. It is there, what you call them, that is like for humans, we call it is fog chal raha hai. Fog is a good scent for us. So same is the fog for animal. So they highly depend on the smell. And we should take care that such kind of smell completely not eradicated when we change their cage. We should always take some of the bedding and put them into the new cage. They are sensitive to ultrasound. Many of the people think animals are dumb. They don't talk. Not you. I can show you a video. You can take my number and my phone number and just give me a WhatsApp. I will send you a video. They talk to them, but they talk to whom? They talk in the range of voice, ultrasonic range, which we don't understand and which we cannot hear. So, if I am going and only causing problem by giving injections to them, they might be talking to each other and saying, oh, there is a men have come because they understand us by smell and they will tell us, oh, I have been given injection, now it's your turn. So these are the parameter, like they are the most master digger. Master, they want to dig. We cannot provide them all these conditions, what they need into natural environment, but we can provide them some of the condition by tissue enrichment, tissue paper enrichment, or some of the enrichment like wheels and some other things. by changing them so that they have something to be done. So they have the mental control. They are not feeling bored and they don't have the stereotypic environment. And they are, when you handle them, don't handle them by tail because that makes them extremely anxious. Handle them by the cupping, which is a very new technique nowadays to handle. And their most comfortable and temperature is 26 to 30. So what they want, we have to provide them. How do you ask them? If you ask them, what do I want? I will tell them, I want this. I want dal bati churma and XYZ. But how do you ask animal why they want? Okay? It's not very difficult. How do you read their mind? Just do the preference test. Okay? Now, what is the preference test? Here you can see the two case. There is a connection between the two case. In one case, you provide X thing. Another case, you provide Y thing. So, and you will see, they will spend most of the time where they like. They will take whatever it is being liked by them. And that is how you can ask them what is liked by them. How can you talk to them? By giving preference test, by giving and correcting two case, and also by giving different condition. Like in one case, you put light on, another case, you don't put light. So you can see whether they prefer light or don't prefer light. In one case, you provide enrichment tissue paper. In another case, you don't provide enrichment. So you can say, oh, they don't like enrichment or they like enrichment. In that way, you can read their mind, you can ask the question, and you can try to provide the same. So this is what we have already discussed, the preference test, which is being done by connecting the two case and providing the different case. Another thing that I want to emphasize is the unrelieved pain. Most of the time, we consider animals don't feel pain, which is not true. It is our stupidity that we don't understand their sign of pain. They understand pain, but we don't understand how to interpret the pain. We always think it is as like we express, the answer express, not you. So animals do not understand the source of pain. Why it is important to unrelieve the pain. I have pain now. I may anticipate that I will go to the hospital, and by the time I will reach to the hospital, I have suffering till the time. Otherwise, I will be given injection, and my pain will be relieved. But animals cannot do that. They cannot rationalize that it will end soon, and their whole life becomes pain. So, for moral and scientific reason, one should expect a crucial emphasis on pain control. Whenever you are doing any procedure, you have to control pain. Same pain killer which are used for humans can be also used in animal. That you have to ask your veterinarian. But any procedure which causes pain in animal also causes pain in human. Any animal which is showing repetitive behavior is suffering. Animal usually, mouse, how do you know that animal is happy and healthy? How do you know I am happy and healthy? I am now taken bath, clean and neat. But mouse spend 80% of their energy in cleaning themselves. So you will find a spotlessly clean mouse. It means mouse is happy. If you find a mouse in dirty and not very well clean, it means mouse is in pain. Okay, so here you see the example. One animal is in pain, another animal is not in pain. So besides the scientific benefit, there are other advantages associated with providing enrichment. Giving an animal item also give them additional means of communicating their state of well-being or whether they are suffering. On the right side, you see animal is not means interested and it is defecating everywhere. Where on the right side, animal have made proper house, they have made proper nest, and also he is very alert. He is not alert. He is not showing any interest and just passing time. So this animal is in pain. This is not in pain. Here you can also see the difference. So in this way, if the nest is formed or nest is not formed is also indicator whether animal is in pain. Here is very good example. On the right side, you see, you gave some tissue paper. On the right side, you see a lot of tissue paper is cut into the pieces. It means he is making its own house. He is happy. You see, animal is not doing anything, not showing any interest. Animal is in pain. So these indicator of nest calling, burrowing behavior is a clear sign that animal are suffering, animal is in pain. So you, when you plan your experiment, must take care of all these things. pain or no pain. Now comes the question of what the law says. You have to do an experiment, and it is by law, it is mandated by law. You have to take ethical approval, and after ethical approval, you have to start. And by law, we are bound to treat animal compassionately, as per the I told you section 51A of the Constitution, and also prevention of cruelty to animals says we have to always avoid unnecessary suffering. Okay, so when you do experiment, how do you know whether suffering is necessary or unnecessary? So I give you one example. So there was one farmer who was bringing his cattle for slaughtering from X place to Y place. Okay, so what he did, all the cattle were having the horns. So he removed the horns, he chopped off the horns, and he took the cattle to the slaughterhouse. And when they reached to the slaughterhouse, the doctor said, "Hey, you have caused necessary suffering." Police said, "Necessary suffering." Doctor said, "Necessary." He said, farmer said, "No, it is not necessary. It is unnecessary suffering." Okay. Then it went to the court. Court asked few questions, and that you have to ask these three questions. Farmer said, "I have brought these cattle from X place to Y place, and I have removed the horns, because if you don't remove the horns, they will fight into the truck and they cause injury to them. Hence, it is a good thing I have done." Another thing, earlier I could only pack 4 cattle, now I can put 10 cattle. So it is good for me, profitable for me. So it is good for animal also. What do you think? So for thinking, you have to ask 3 questions. Okay, this is the Ford and Wiley case 8829. If you want, I can also share with you. So basis of animal practice is made up of these 3 components. Our prevention of cruelty to animals is unnecessary suffering. Now, question number 1, what we have to ask: did the animal suffer? Yes, if you take out the horns, chop off the horns, animal suffer. It is like cutting our finger. Oh, yes. Was the suffering necessary or it could be avoided? In my sense, you also take, it could be avoided. You can pack lesser number of animal, it could be avoided. Would a reasonable, competent, reasonable human farmer have tolerate such a state of suffering? Because in law, if you are not reasonable, if you are mad, if you are proven not mentally sound, no laws apply on you. Okay? So, would a reasonably competent, reasonably human farmer have tolerated such a state of affair? No. Okay? So, if you are doing experiment in your facility, you ask whether animal, you cut the tail for taking that piece of tissue for genotyping search, or you take blood from the eyes. Did animal suffer? Yes. Was suffering necessary or unavoidable? Could be avoided. Would a reasonable? How could you avoid? Here is the trick. You could take the blood, but from not the eye, but from the tail. Not from the eye, but from the mandible. From the eye, yes, but with anesthesia. Suffering is avoided. Suffering is, did the animal suffer? No. Why did not suffer? Anesthesia. Suffering could be avoided, could not, because you are doing experiment. But it is avoided by anesthesia and analgesia. Would a reasonable, competent, reasonable human farmer or human researcher? Yes. Because we are avoiding suffering, and suffering is necessary, because whatever comes out from the research will be more useful for avoiding suffering to the human. So, what we call that we have to balance suffering and benefit. Okay. Now with this you, I think you can do very well your these things, whether the suffering was necessary, suffering, necessity, and mental elements, mensera, because all of you are doing PhD or master, it means you are mentally sound. The farmer is also mentally sound, because he is talking about profit. He is also talking about backing the animal. So please do this formula for your experiment, just to say whether it is necessary or necessary. Usually, when you are doing experiment on human, You ask humans, can I do experiment on you? Now you must be hearing the corona vaccine. So you ask volunteer, who can volunteer? So they will volunteer, I will volunteer, I can undergo this agony for the suffering to the humans. But animal cannot because they cannot speak. Who speaks in their behalf? We speaks. If you speaks on their behalf, because animal cannot speak, you are doing experiment on animal, who is their advocate? It is the researcher who is the advocate who speaks on behalf of animal. Okay, so you must take care the pain is not there. So what are the issues in experiments? There is a lot of failure rate. You can see this graph where from preclinical phase 1, phase 2, phase 3 and registration, the success rate is low. So sometimes we are being criticized by the that we are experiment failure is a lot and we are not doing right experiment. But here there is a point. If you see the preclinical to the phase 1, preclinical to the phase 1, preclinical is done on mouse or anyone, and phase 1, phase 2 done in humans. So preclinical to phase 1, there is hardly a toxicity. Toxicity is very less thanks to the animal. Otherwise, if there is no animal testing, all this toxicity will go to whom? Us, and we will die. Second thing, animal experiment fail, yes, but not animal experiment. Human experiment also fail. You say phase 1, phase 2, phase 3. Here also the failure rate is steeply going down. So we have to do experiment correctly. How do you do experiment correctly? You have to choose the right model of your disease. Suppose if you are doing experiment in diabetes, animals are different from us at nucleic acid level, protein level, pathway level, cell level, tissue level, organism level, organ and population environment. They are totally different. And we should aware of the difference. Like I told you, the tissue difference. The glucose clearance in mouse is liver, glucose clearance in human is skeletal muscle. The receptor and how do we eat? We eat breakfast, lunch, dinner. How do animal eat? They always keeps on eating. How do the insulin secrete in animal? Totally different from insulin secreted into us. We take breakfast, we take milk, we take something sugary. So a lot of glucose into our stream, and there is a secretion of insulin. There is a hyperglycemia, then it comes to the normal level. But no, animals usually keeps on eating, keeps on eating, keeps on. So that their secretion of insulin is different from ours. You should know these differences, and then you plan your experiment. So species selection of your animal model for your research is extremely important. It is most based on your ability. When we sit in ethics summit, usually people say it is available in our university, it is cheap, it is breeding very well, it is easy to handle, their breeding cycle is very fast, so I can do it. No. You have to tell what is my research, what I am researching on. This is the thing, and what is this matching into animal model. Okay? Then only you can say your model is correct rather than availability. Why most published research findings are false? Another example is that I tell you that biologists are not good in statistics and statisticians are not good in biology. So today's organizer, Professor Anjali, is the professor of physics, I suppose, or I don't know. Yeah. But biologist, for a biologist, it is very difficult to understand statistics. I'm a vet. For me, also statistics is different. So here the p-value is less than 0.005 and significance is done. That does not indicate the power of your experiment, reproducibility of experiment. So it is important when you do experiment, you should understand the sample size. What do we do sample size? How many animal in a group? 6 number of animal in a group. What is the base? I don't know. Usually say to get statistically significant. No, to calculate the sample size, please you have to understand the alpha, beta, signal, and the standard error or the variation. If you know all these parameters, then only you can calculate the sample size. If you are doing regulatory experiment, it's okay. What the regulation says, you have to follow. But if you are doing experiment in your lab for something new, you have to calculate sample size. If you cannot calculate some precise anything because there is no standard deviation or this data is available, you can go for resource equation where the number of animals usually come to 10 in a group. So do give attention to the sample size. So why do we give attention to the sample size in this experiment? So there is a systematic review and meta-analysis system to know that which way we are going. And there is one example I quote, which is about the stroke. There 1,026 drugs were tested for stroke in animal models, and only 374 were found effective in animal model. 1,026 and 300. So instead of animal model, this is tested onto this cell line. But 370, sorry for this correction, I mean a mistake. 374 found ineffective in animal model, and only one drug was found effective in humans. That was also the new drug. That was the repurposed drug. So you see how animal confusing the data are, which are published already in the public domain. So systematic review and meta-analysis, if your disease of interest is the best way to see which animal model is correct, one model to be selected. So at the means I'm coming to the last because 5 minutes only is left. When you are in doubt, support the animal. Can we really call ourselves animal carer when we allow mechanization and complacency? Please, when you are doing experiment, don't say complacency. You have to be critical of your experiment. Discuss in somebody, discuss in your group, and as much as possible. If you are very say, oh, I know you, you know me, and you don't criticize, that's not a good way. So in that way, we cannot call ourselves animal carer because we are allowing mechanized and complacency. And what is in other context is we are perpetuating nothing but cruelty. Tradition and status quo is another problem. We are always doing in that way. We are this department running 600 years or 80 years, and we are continuously doing it. We don't want to change. Resistance to change, both individually as in the form of institutional inertia, all combines to challenge those who ask epidemiological right questions. So, science is changing day by day. Things are changing, and that's why because of this we have a ethics committee. It's important to adopt these things. Another thing, how do you report these animal experiments? Don't write something which is politically correct. Like here, people are saying great care. What do you mean by great care? Great care means just go and visit animal facility once a day and just provide the feed and water. That's great care? No. Great care is not that. You have to quantify the great care. Great care has gone into refining the techniques used to monitor animal response to the treatment using order to reduce the degree and duration of what is degree and what duration of suffering incurred to the minimum work with mice. Cattle is not expected to be greater care than moderate severity. Experienced observer. Who are experienced observer? Those who are trained and certified, those who are veterinarian. The veterinarian who had worked into animal husbandry for whole the life is not experienced in laboratory animal science. Okay, experience means something who is certified. And all time monitor clinical sign. Which clinical sign? Here you may sound the very good care, good words are like humanly. What do you by humanly? We are all human. Humanly means you go out and help positively. So in this one, they have not mentioned what is the intervention, what is the frequency, what is the duration, what is the adverse effect. Yes, they have mentioned severity and also the pain. So here the chapter is very good. English is very good, but it is of no use. But small chapter can give use. Here you can see the first example. Basis of experiment is surgical closure of the middle cerebral artery. They are not saying good care. They are saying they are closing what? Middle cerebral artery. What will happen if you close and how you are closing? By under anesthesia. They did not mention anesthesia. They said great care. This result in mice a stroke which is manifest. What will happen if you do this? Stroke will happen. What will happen in stroke? Hemiplegia. Means one side of mice will be paralyzed. Sorry. Which constitutes the main factor and is classified as medium severity. The feed intake is mice may take the feed. The observation time is 90% of animal for 24 hours. So first 24 hours you are observing 90%. Means you are there for 24 hours. Observer. Who observer? Experienced observer. Who is experienced? certified. And after 24 hours, 10% for next 72 hours. So here they have quantified everything. They have not used humanly. They have used term anesthesia and analgesia. They have given time. They have given what will happen. They have given what they will do. So they have given intervention, that is middle cerebral artery, frequency 24 hours, 72 hours, duration they have given, adverse effect, hemiplegia, severity, very high severe fate, what will happen? This paraplegia or hemiplegia. So this is science. Instead of writing great care, humanely, these experienced observers, they carry no importance. You have to quantify this. Ultimately, you have to do the cost and benefit analysis. Cost is the suffering to the animal. What is the cost? Is suffering to the animal. What is the benefit? Benefit is the research output, and which will be helpful for the humans or for the animal. Cost is suffering. Who is suffering? Animal are suffering. Benefit is to whom? Any humans. When and how. So this is a very debatable. So we have published several publications, and recently also we have published a systematic review. All these concepts are given. You just go and see the PubMed and type our name. This is the sample size I already talked about. You have to know alpha, you have to know beta, and you have to also know the signal. What is signal? Signal is something that we are expecting. If we are taking, suppose we have our drug to be effective for bringing down the temperature. Temperature means fever, 102 to 100. So from 102, my paracetamol or my drug will bring down the temperature from 102 to 100. So the signal effect size is what? 2 degrees. From 102 to 100 in that way. And also the noise. Noise is the intrinsic variability. The standard deviation or the variation which is there always, we have to find out that. So this is somehow you have to calculate the sample size. You have to standardize your animal experiment. You have to say minimum variation, and you have to standardize. You have to give same water, same feed, same bedding, same light, same animal handler also. So environmental condition, experimental procedure have to be same. And this will really help into reduction of bias, number of animal needed, and also enhancement of reproducive result, animal welfare, and quality of research. So the basically we have to amplify by all these procedure. We amplify what? Signal. What is signal? I told you that what we expect. What we expect is that if you are doing research for parasite, the temperature bring down or a cancer, it will reduce the cancer from this level to this level. What is noise? Noise is that you have a temperature control today. Tomorrow you don't have a light in animal facility. You have feed today from X supplier. Today you have from X supplier. Today your experiment is done by Vijay Singh. Tomorrow you are Paul Singh. Then tomorrow you ask your madam Anjali that can you do experiment? These all things bring noise. So if you have a noise and signal equal, no statistical significance, hence no useful data. Okay, so reduce the noise, amplify the signal is our purpose. How do you do amplify the signal? By doing standardization. Noise also by standardization. Signal can be amplified by using animal which is right animal. Okay, so these are the some of the variation which is often more than what we think. Genetic makeup of the animal. Don't say every white animal is Walpsy animal. You have to do the genetic monitoring, environmental condition, biological effect like infection, case density. Sometimes animal is perfectly healthy, but they have hepatitis or mycoplasma, which is very common in our part of the world. So you have to take care of the temperature, lighting, infection, case density, humidity, noise. Randomization. Randomization means not haphazard. Whatever we do in a staggering way. So because of the time paucity, I cannot cover all these things. And also the chemical which may come from the bedding material. Suppose if a farmer is spraying pesticide onto the in this rice husk or rice field, and you are taking rice husk as a bedding material. So this pesticide is coming to your experiment, which you may not be aware. Okay, so these things are the most some of the common error which make is the type 2 error, where the effect is small, variation is large, and number of animal used is very small. So we may Sample size is that too few animal is a complete waste, a total waste, and too many animal is a partial waste. But best is to calculate the sample size. We also have to justify the animal usage with the cost and benefit analysis. You cannot justify just by saying that it is by for my PhD, and you have to be always critical what you do and don't believe in status quo. So I hope I have cleared some of these doubts. Animal welfare is different from animal protection. Well, necessary is different from unnecessary. Ethics is different from welfare. So this you can discuss later on because of the paucity of time. And we should focus on systematic review and meta-analysis, specialized animal training, statistics, experimental design, animal welfare, cost and benefit analysis necessary, and intensity and duration of pay. Hey, so at last, I have a message to all of you. Hey, human, be human. This is the photo of my IGIB, where the last time I'm continuously doing 2-week training program on laboratory animal science, and Dr. Kumud, myself have done several programs on National Institute of Animal Welfare, and throughout the country, nook and corner, we have trained the person to do ethical treatment. This is about my contact detail in case if you need. You can always contact me. And with this, I would like to thank for the opportunity. Thank you very much.
Thank you, sir. Thank you so much for such a wonderful lecture you delivered today. You have explained all the basic concepts of animal behavior, animal handling very well, and you addressed different examples, common examples. That was the best part of your presentation. And I hope, and I can see as well, very good feedback from the participants, that all the participants, even including me, must have enriched their knowledge. They must have been benefited through your lecture. So, sir, can we take a few questions from the participants?
Yes, please.
Thank you. The first question is from Mr. Anurag Mathur. What is potential role of animal wildlife welfare in environmental legislation?
Can you come again, Dr. Anjali?
Sure, sir. What is potential role of animal welfare in environmental legislation?
Yeah, animal welfare, I told you, is something. What was the questioner name?
Anurag Mathur.
Mr. Anurag, animal welfare, whatever the animal, either wild animal or domestic animal or whosoever it is, the welfare is something which is the natural need and of the animal, what they want. If you are keeping animal, suppose wild animal, keeping into the captivity and breeding and doing some experiment, like we are seeing so many diseases coming from animal. I quote example, I'm a vet. 60 to 70% of these viruses and diseases are jumping from whom? Animal source. So their welfare is important, our welfare is important. Conservation environment is only possible when we maintain the diversity. Nowadays, this is very pertinent question, and I can speak on this a lot. We are removing the biodiversity and promoting more monoculture, more laboratory based, which is not really true and which is not really good. And because of this is our kind of a mechanical thinking that this virus keeps on jumping from species to species first. Conservation is not possible. So I give one example which I have experienced. If you house a monkey for a very long time into a laboratory, and suddenly you feel, no, you let them loose, let them go into the wild. Can they survive? No, they cannot survive. Because they have forgot their natural skill to survive into the wildlife. So this welfare is really important for conservation, but the welfare and conservation can only happen when you treat them in the way they are. And for understanding that, we have to understand their behavior. And their behavior, first we have to understand that anything which is good for us is not good for them. This is the most central problem we feel. Air condition is good for us and air condition is also good for them. This is good for us and it is good for them. No, that's not true. So we have to really understand, and to understand that, I quote you an example of doing preferential test. Understand their mind by ethological test. And I can quote many examples, but this is in short, Dr. Anjali.
Thank you, sir. Thank you for the nice explanation. The next question is from Mr. Avinash. How can I determine reproductive senescence in female laboratory animal with and without dissection?
Reproductive senescence
Senescence in female laboratory animal
with and without.
dissection.
First of all, dissection is not allowed, dear friend, whosoever you are talking about.
Mr. Avinash.
So, Avinash, let me be very clear that dissection is no longer allowed now, either in the frog or in the animal. Now, I told you the first slide was what is an animal experiment? Animal experiment which is done for certain purpose. Dissection is not a purpose, so that's no longer allowed. If you don't have a purpose, and purpose is used to solve some societal problem. So if you want to see, I did not get what you want to ask. Means dissection is not also allowed.
I think he want to know like the reproductive senescence in female laboratory animal. If Mr. Avinash is there on meet, he can ask the question. He can clear out the question.
Senescence, I don't know what he is asking means.
So we can move on to the next question. He can specify it later. Mr. Kulbhushan, are there some health disorders by consumption of broiler chickens?
This is more a question which is I'm doing work now in food safety. So this is nothing related to welfare, but food safety. But in this, I would like to say, if your broiler is grown up fed with antibiotic, okay. I told you the broiler earlier used to grow into the same size into the 12 weeks. Now the broiler is grown into 25 or 30 days. They are sometime coming early into the market. If your broiler is not safe, the food is not safe. So I can only say that you have to know the source of your broiler. If you are getting broiler from some farmer who are feeding something which is for the growth, and which is not good for you, of course, it's not good for you. But it has nothing to do with the welfare, but it has related to food safety. Now, broiler are coming with, you may see some of the advertisement in the newspaper, you may be seeing in Jaipur also. Sometime you get antibiotic-free chicken or antibiotic-free milk.
Right.
It means people are becoming aware that antibiotic is given to the animal. This antibiotic is coming to us. If you will go to the doctor when you will fall sick, this antibiotic will naturally not work in you because you are already exposed through milk to those antibiotics. Okay, this is also a big chain. Then your flora, there is a lot of bacteria which are there that normally into our intestine, they will be changed. So they will not be longer the same. So the nutshell is your food is safe when animal feed is safe. So you ask the question to the farmer from where you are bringing the chicken, whether the chicken was fed with antibiotic or not, or otherwise test it. Test means TST, test. Test is for the presence of antibiotic residue. Right. Then only it is safe.
Right, sir.
Nice question.
So one question in the pipeline I also want to raise. Like when we feed, like we use these like the broiler which you were discussing, or any vegetable which is off season or which is not grown in that particular area. So when we consume that, so I think the stress related proteins might be present in that organism. So when we are consuming those same stress related proteins are consumed by us. So is it the cause of persisting stress in humans nowadays? The raised cause of one of the cause maybe.
Yeah, because now we are coming animal to the plant, but basically this is a food safety issue again, Anjali. What we propose that we always eat what is local and what is seasonal. This is the mantra, what we say in food safety, rather than bringing something from somewhere, because the demand which our body needs is as per the local, it adjusted as per different local conditions.
Exactly.
So we always say in food safety that eat local and also eat seasonal. Something which is fashionable and very costly is not always good for your health.
Right, sir.
That I can say.
Thank you, sir. Sir, we can take two more questions. One is from Miss Shreya. How to determine sample size?
This is very, very good question, Shreya. And I think because usually when I do, it means not virtual class and real class, I ask student to do it. Because whenever you go for the animal experiment, usually people say, how many animals? 6 number of animals. Why you take 6 number of animals? Because we want statistically significant. No, that's not true. Sample size means you have to determine your alpha, which is usually 0.05. You have to determine your beta. Beta is the power that your real effect is detected. Your real effect is covered into that, and that usually you take 80 to 90%. If you go in Google and collect the sample size, next is the effect size. Effect size is what you determine your animal experiment. What do you want to do? Suppose if you can see my video, you are discovering a drug for cancer, and you are discovering that cancer size of the tumor is by vernier caliper. You say this is the size, and you determined by this is the 5 centimeter size. Suppose this is the 5 centimeter size. I want my drug to reduce from this 5 centimeter to 2 centimeter. So effect size is from 5 to 2. So effect size is 3 centimeter. So you have to determine your effect size, and you also have to find out your variation, standard deviation, or the noise we call in simple terms. Noise can be found out by already published data. So when you have all these things, alpha, beta, effect size, and also the variation or noise, you can determine your sample size. But it is important to determine the sample size. Don't go for 6. And usually we always go for 6, which is not correct. And nowadays reviewers are also asking, how did you find out this is your sample size? So you have to, and whoever means ask this question, please go to the Google, write sample size calculation, determine your alpha, beta, effect size, variation, or noise. Then you can easily calculate. And that for that, you have to read a lot, because for finding variation and noise, you have to read the literature. If you want to detect your effect size, you should already know what is already in the market and what you want to determine. So it's not easy. And that's why people take shortcut and say, right, 6.
It's easy.
Magic number.
It's easy, sir. So the last question, it's from Miss Varsha Dikshit. Which model animal is best for diabetic studies?
This is not, I cannot give the tailor-made answer to this, Varsha. But this is good question. Varsha, for finding out the answer to this, first go and sit with a human doctor. This is my personal suggestion. What they see in human, when a diabetes patient go to a human doctor, what they will see? They will see glucose, and they also will see the glycosylated hemoglobin nowadays, which is indicator for diabetes syndrome. If you find these two things together, you can say the model is correct. First. Second thing, that when you are selecting a model, you don't have to select based on what is available in your university or your college. No. You have to understand what is happening in humans, what is happening into animals. Second thing, how diabetes is happening in humans, not by giving streptozotocin injection, that is type 1 diabetes, because you have destroyed beta cell. No, that's not good model. How diabetes is happening in humans, we eat a lot, we don't do anything, we become docile, and usually by the age of 40, 45, we are obese, we are having hypertension, we have other syndrome, and ultimately we become a model for metabolic syndrome, and we also have a diabetes. Try to have same situation in mouse. Feed them mouse high-fat diet, create them obese, so that also they become a diabetes in a natural way, rather than giving streptozotocin injection, which destroys the beta cell, and which is a model for hypoglycemia, but not a model for diabetes. But not a tailor-made answer for any question. You have to read a lot.
Right.
This is what I can suggest you for diabetes.
Thank you, sir. Thank you for your suggestions and sorting out the queries raised by the participants. May I request Professor PJ John, sir, if he is there on Meet, to address the participants and our speaker as well. Dr. PJ John, he is Head Department of Zoology, University of Rajasthan.
Great, sir.
Sir, can you hear me, sir? Are you available? Okay, I'm sorry. I think.
I can. No, I can see him, but he's put this speaker not off. Speech is off.
He's
You
on
ask him to mute mode.
mute mode, right, sir?
I can see him. John play. Yeah, Dr. John is there, but he's speaking, but it's in mute mode.
Acha, he's speaking, but he's sir.
He's speaking.
Okay, sir.
You can call him by.
Exactly. Thank you, sir. I'm just.
Yeah.
Okay, sir. We are unable to connect to him. Anyway, sir.
Hello.
Yes, sir.
Yeah, we can hear it.
Hello.
Yes, sir. Sir,
Hello.
Hello. Yes, sir. We can hear you.
Can you hear?
Yes,
Can
sir.
you hear me?
Yes.
Yes, sir. We can hear you. John, sir, we can hear you.
Hello.
Hello.
Can you hear me?
Yes, sir. We can hear you.
Yes.
You can hear me?
Yes, sir. We
Yes.
can.
Okay. Sir, first of all, I would like to express my sincere thanks to Dr. Vijay Singh, sir, who has spared your valuable time for us and for the participants. And really, sir, it was a wonderful lecture and very informative. And very what I should say, even some of the queries which had in my mind for a long time has been cleared right now. Though maybe on the verge of calculating all my research and everything, as maybe these 2 years. But the youngsters who are just started their research as well, for them it is really an informative lecture, sir. I'm really thankful and I'm really glad to have you sometime in our department in person to see how to guide us because that would be a wonderful thing because of course the online lecture we all had many more questions and queries in our mind to validate to expedite our experiment in a better way. So that is my personal interest to have you in my department. Of course, even the situation is getting into normal. So anyway, sir, I congratulate you and I thank you once again, sir. And all here my participants, I really express my thanks and gratitude to all you have made it possible to be part of this webinar. And of course, the whole age goes to Dr. Ashley Abasi, my colleague and good, friend. And he took all the efforts to make great success. And with his chief, Dr. Kamlesh Awasthi, from Diabetic department, Dr. Kumud Awasthi from Vivekananda Global University. Excellent job, and put it because they are the one who made go ahead. So I'm also happy that also I can depend on people like Dr. Kumud and companies and people and like my department can only fun and everyone in my department can excellent and thank also giving a lot of support, not only moral support, but by scientific advice and
Thank you, sir. Thank you for your kind words. You also know that it won't be possible without your support and encouragement to conduct this webinar. Thank you, sir, Dr. Vijay Singh, sir. Once again, I would like to thank you to deliver such an informative lecture. Sir, you can drop the call from your side.
Yeah, I would like to thank Kumud also because we have worked together a lot and he's my good friend. I don't know whether he can hear me or not.
So
and
he's
together
available.
we have developed so many good modules. So thank you very much, Anjali, for giving me the opportunity. And I just would like to say I'm always available and without cost. Even I can pay you. We are doing all these conferences, courses all over the world, not India.
Okay.
Like we have done world like in Malaysia,
Okay.
Vietnam, Sri Lanka.
So we can plan
where
something, sir. We
traveling
will
all over.
discuss it over it. We will discuss over it
Yeah.
certainly shortly.
Thank you.
Okay. Thank you, sir.
Namaste.
It was our pleasure. It was our pleasure, sir. Thank you,
Namaste.
sir. Thank you. Namaste,
Namaste.
sir. Dear participants, so this was the last lecture of this webinar. Uh, I thank you all. I thank you all very much for joining this webinar. I tried at my end to connect with you, to bring the experts from different areas. I was able to bring experts from plant molecular biology, from animal field, uh, and I know from your feedback, from your comments, that your knowledge might have been updated and enhanced with the topic. I thank you to those also who could be benefited and appreciated our efforts in this difficult time. You know, we had limited resources. There was initially there was internet issue. We had some problems during quiz preparation. We need time. There was a number constraint to incorporate people on meet. So we tried to resolve these issues each day. Even like it was my first experience, our team conducted this kind of webinar for the first time, and we are also in the learning phase, and we were also able to learn many new things. Great experience. I would like to give a big thanks to our critics. You know, without whom help it was not able to, we won't have been improved our deliveries to you. And one thing I want to say regarding certificates, this was the highest rated query in each and every session about the queries, and post session as well. Like now I would request you to give some time to us, a week time, and then we will send you certificates, e-certificates on your email after a week, since we have to resolve certain issues like each day on meet and on YouTube. We have around 200 participants. Like today also we have 210 participants, but at the end of a quiz session the responses which we get, they are around 280, 290, and we find email addresses like abc@gmail.com, papa@gmail.com in the response box. So I don't know, like they are not the registered candidates, of course. And you know, we are able to identify those persons as well, but it's not good to expose their name here. So intentions were like good, to learn. Yes, I think to get a certificate must be a secondary thing as compared to get knowledge. So once again I would like to thank you all, and the link will be shared shortly on meet as well as on YouTube. And let's hope that this difficult time it ends soon and we all get back to normal life again. And I would ask you all to stay home, stay safe, and learn online these days. Thank you.